our Habitats and Ecosystems session. We've got 2 talks in that session and then a fish and fisheries talk before our break. So our first speaker is Jezreel Wilson, and her talk is Fungi on the Edge, Ectomycorrhizal Diversity in South Texas Prairie Dunes. This goes forward.
Cool. Everyone, my name is Jezreel Wilson, and I am your presenter, and I will talk about fungi in the coastal dunes. I'm from A&M University, Corpus Christi, and I am a second-year master's student. So fungi on the edge, ectomycorrhizal diversity in the South Texas prairie dunes. So a little bit of background. So the South Texas prairie dunes, this is an ecoregion, and it's pretty unique. It's really beautiful, and it stretches across barrier islands and inland zones. But it's not just really beautiful, it is really important for coastal defenses. It's a natural buffer against hurricanes, storm surge, and sea level rise, and it stabilizes our inland communities. So as you can imagine, the life that has to thrive here, whether microbial or vegetation or fauna, it has to withstand these environmental pressures against hurricanes, sea level rise, and storm surge. And among the life that thrives here are
You can use the space bar as well if you need.
Okay, among the life that thrives here are spoiler alert, okay. Among the life that thrives here are soil microbes. So below your feet, living within the plant communities are these soil microbes. And among those soil microbes, if you didn't already know, are fungi. So you have your soil microbial bacteria, but among them are the mycobiome, kind of adjacent to the microbiome, but the mycobiome is the fungi. So, and then among those fungi are ectomycorrhizal fungi, and these are fungi that do not penetrate inside the plant root cortex. It's ecto, so it's the outermost part of the plant roots. And here in blue, they don't penetrate inside the plant cells, but they kind of sit there as a sheath. And what the ectomycorrhizae does is it kind of acts like extra roots for the plant. It helps the plant survive high salinity, drought, heat, and perhaps survive in nutrient-poor soils. So survival here requires some pretty serious adaptations, and perhaps these fungi might be the key to alleviating some of those survival pressures. So let's talk about those environmental pressures. So the first one would be salinity. These areas where the plants grow have to withstand salinity pressures. And then perhaps soil nutrients like soil nitrogen and soil carbon that are essential for plants. And then maybe it's site-specific differences. So maybe in one barrier island, there's specific microhabitats suitable for plants that might not be found in another microhabitat. And so despite all of this, and despite the importance, we know pretty little about ectomycorrhizae in the coastal dunes. A lot of the literature has been studying ectomycorrhizae in forested areas since the 1800s, but little is known about how they behave in the coasts. So what I aim to uncover is how diverse are ectomycorrhizae, kind of an understudied group of fungi in South Texas coastal prairie dunes, and what are these factors that are influencing the community composition? So who are they, how diverse are they, and what are the factors shaping their communities? Perhaps it's soil nutrients, perhaps it's salinity, perhaps it's site-specific factors. So what I hypothesize is maybe that the overall diversity patterns of the soil microbiome are exhibiting some kind of soil, spatial structure. and then maybe link to some of these soil properties. So what we aim to do is travel to 4 sites. So Mustang Island, Padre Island, Matagorda, and Galveston, just a few. This is still in ongoing research. And then our sampling protocol is we took one sampling site, which is 30 meters by 30 meters with 9 subsamples, collected 5 to 10 centimeters depth of soil. And then we homogenize the soil samples, and that would be our one site. And then each site is 1 kilometer apart, so we would just hike 1 kilometer apart. And so our methods, we collected the soil samples, we sent them off for libraries to target specifically the ITS2 region of the fungi, and then sent it off for next generation sequencing, and then ran a bunch of analyses. So what did we find? So just kind of some preliminary analyses. What did we find? Site specific. We calculated some alpha diversity metrics. We sequenced DNA and we got ASVs, which are amplicon sequence variants. Amplicon sequence variants are basically the fungal fingerprint, because it's really hard to identify fungi specifically by species. So we went by ASVs, or amplicon sequence variants, short sequences of DNA, if you will. And so what we found was quite interesting. Differences in species richness across these 4 different barrier islands, with Matagorda and Mustang Island showing some differences, and then Matagorda and Padre Island National Seashore showing some differences. And then we also looked at Shannon diversity. So this counts for species evenness and species richness, and we found that Matagorda and Mustang had some differences. But this just shows us a specific snapshot. It just shows us the entire microbiome diversity. So what kind of fungi are there? But we want to focus on ectomycorrhizae, right? Like just one specific type of fungi. But our molecular marker kind of targets lots of fungi. So to do that, we had to look at functional guilds. I had to ask myself, okay, I have taxonomic data, let me assign some functional guilds. So I used FunGuild. So I'm focusing on the actual guild to understand the ecological function. So some types of guilds, some type of ecological functions that fungi have, well, fungi can be pathogens. Fungi can be AMF, arbuscular mycorrhizal fungi. They can penetrate inside the plant root. They can be ericoid, they can be foliar, they can be like innocuous. They can be mycoparasites, plant pathogens, or just your general decomposer. But what I wanted to focus on was just the ectomycorrhizae. So when I used FunGuild, I looked at the entire microbiome in general. So this is just microbiome in general, and you can see the different beautiful colors represent an entire guild. And then looking across the 4 barrier islands, you can see the more color there are, the more of that specific fungal guild there are. As you can see here in this beautiful light blue, there's lots of animal parasites relatively in each of these barrier islands, and then animal pathogens. And then of course you've got your undefined saprotrophs right here in the purple. But what I'm focused more on is the ectomycorrhizal part. So lots of guilds, but I want to focus just on that one particular part, which is, it looks like it's relatively absent here, but they make up a small part in the entire microbiome. So going back, want to focus on the ectomycorrhizal guild only. So then I had to filter for the ectomycorrhizal guild. So focusing on just that one particular ecological function, because when you amplify the ITS2 region, you can target non-ectomycorrhizae. This is the ITS sequence. So then going back to our functional guilds, I just want to filter for ectomycorrhizae only. And then when we do that, we get something with fewer colors. Looking at just families per site, as you can see, some sites are dominated just by one ectomycorrhizal family. This is just based on the UNITE database, however. And then there's some islands that have a little bit more families. I'm still trying to analyze this through genus, but that will require looking through lots of literature because some of these mycorrhizae actually are not categorized through genus. Like the highest resolution I can get is the family. So that will require digging through literature and figuring out what has been identified to a genus or species level. So for now, this is the highest resolution, is through family. We can see here there are some sites that are just dominated by one ECM family. Especially these two causes. And then I also created a Bray-Curtis similarity matrix. So I wanted to quantify if the different ECM communities across the samples, based on the ASVs, based on the amplicon sequence variants, how similar each pair of samples are, how different one soil sample is from the other. And then I tested whether salinity, nitrogen, or carbon were significant predictors. And then yeah, we plotted them, and as you can see here, the samples are clustering by island, so indicating some strong site level structure. And then perhaps salinity and nitrogen are significant predictors, but I am adding more factors into this. And then salinity likely explains some shifts in these dominant ectomycorrhizal taxa that we're seeing here. So, so diversity of ectomycorrhizae so far in the coastal prairie dunes in this vital ecoregion. We looked at the diversity metrics. And then from the entire microbiome diversity, I filtered for a specific guild for ECM. If I wanted to filter for another guild, you totally could. And then we just preliminary analyses, we looked at this clustering by island. So all of that to say, these results are showing that they're not just randomly assembling, but there's possible site level environmental conditions, possibly salinity emerging as a key filter. And ectomycorrhizae, I know, has been really extensively studied in forest communities, but in coastal communities, perhaps not so much. So future directions for this, I really want to look at soil pH. I know soil pH has microbiome, has an influence in the microbiome, but I wanted to see if it had an influence in the mycobiome. And then I have another data set, which is the arbuscular mycorrhizal fungi. This is the fungi that penetrates inside the plant roots, but they're not parasitic. I want to look at that and then compare AMF with the ECM data, and yeah, hopefully give you a bigger picture in the future about mycorrhizae in our coastal region. And then I just wanted to acknowledge everyone in the lab for hiking kilometers and kilometers with me, collecting fungal samples, and Society for the Protection of the Underground Networks, or SPUN, for funding the project, and Botanical Gardens for allowing us to go into botanical gardens and literally talking about fungi, kind of taking it over. And there is still a gap of knowledge in coastal fungi, particularly ECM. They are obligate symbionts. They associate with over 80% of land plants, but this is just a start to the bigger picture of how we can conserve them and quantify them. And I think the first step in conserving them is just to see what's there. So.
All right, we've got time for questions.
So why didn't you include plants here? So you only talked about the microbiome. Why didn't you include plants in this study?
So in these, so the question was, did I look at plants and quantify the plants also? So in these coastal dunes, a lot of the plants were like in Poaceae, so like grasses, and lots of Spartina and So we took a general overview of the vegetation that's there, but not specifically to a species level, just what kind of grass was there. So whether it's like Salicornia or something like that. It's just a general overview of the vegetation that's there. But that is something, again, like something that should be looked at.
Do you, so you found some pretty distinct differences at, what was it, the family level between sites? Do you have an idea of the functional difference between those families, and does this have implications for nutrient cycling?
That is actually next. Like this weekend, that is my goal, actually. Because I got these results kind of like a couple of days ago. It's still ongoing, and that is actually the plan for this weekend. So hopefully I can present to you again and give you a better understanding of the different site level, family functions at that level.
Can you tell me about this process of using ASVs to measure diversity and what kind of quality control you did to make sure that those ASVs largely tied back to actual species on the ground?
Oh, the amplicon sequence variant? Oh, okay. So ASVs are, we use a database. So it's a fungal database. It's called Unite. And that's like just fungi. And then you can either compare those to a general fungi database or like eukaryote database. And I just decided to do fungi. And then there is a pipeline for that. This might be, my PI helped me with the entire pipeline. This might be a question, like we can talk about it like afterwards because I have an entire pipeline I like talk to you about.
So, but all of those ASVs in some way were linked back to
Yes.
a fungal sequence.
Yes, yes, yes.
Very nice. We got time for one more. Yeah.
So did you include other nutrients like phosphorus?
Other nutrients like phosphorus, yeah, good question. So carbon and nitrogen were the first ones, but phosphorus definitely has an effect on the AMF, especially, the AMF, our muscular mycorrhizal fungi. So when I study the AMF, that could probably be a good nutrient to look at.
Thank you very much. We'll start up again with another habitats and ecosystems session. Our first speaker is Alexandra Austin, and her title is Blue Carbon Baseline Assessment at Fort Bay, Texas, Study of Stored Organic Carbon in Texas Gulf Coast. Oh, yeah, that's something. You're doing great.
All right, good morning, everybody. I'm Alex Austin. I'm Environmental Project Manager at HDR Engineering here in Corpus. And today I'm going to talk about a blue carbon project that I had the privilege of working on with the Nature Conservancy last year. So for this presentation, brief overview of blue carbon, and then sort of get into what we did on the project and where it actually ended up. So I think there's been a few blue carbon presentations. We won't spend too much time on that, but when we say blue carbon, we're specifically talking about the carbon that's stored in coastal ecosystems. So mangrove marsh, seagrass meadows, and salt marsh, which is what we had at the Fort Bay Ranch. So these wetlands are really powerful carbon sinks. They sequester and store many, many times more carbon than terrestrial systems, but they're also disappearing at a rapid rate. So when they're degraded or destroyed or developed, all that greenhouse gas that has been stored for many, many, many years is released, contributes to global warming, climate change. We don't want that. So the idea behind nature-based carbon credits, of which blue carbon is one type, you can put a value on these, the carbon that's stored on the ecosystem service that the wetlands are providing, and that helps protect all the other services they're providing. So you go from a project, you have a site you want to protect, you have to quantify how much carbon is in it. So it's not just soil carbon, it's what's in the vegetation, it's also what's being emitted to get a total picture of how much carbon is on this site. So you have your site, you've quantified it, you convert that to credits, and then those are sold on the voluntary market. And individuals or organizations can buy those to offset their own emissions. So really it's just a way to protect these wetlands long-term and allow people to make some money off of them. So with this project, we're looking at the Port Bay Ranch. This is in Aransas County. You can see it on the map. It's a 1,200-acre working cattle ranch, right on the shores of Port Bay. So TNC's goal with this was to do a baseline carbon assessment to see if this site would be viable for even entering into the voluntary market. It's a very involved process. So this was kind of a final step after lots of work to get here to identify which site and move forward. So our goal at HDR was to collect baseline soil carbon data, just the soil, and the wetlands and the uplands where we expect that tidal marsh to migrate over time under sea level rise. And then sort of a background overarching goal was just to build the body of knowledge on blue carbon in Texas. This is still an emerging field, and having more people that can do the work is in everybody's interest. So for this project, what we needed to do first was set up a project work plan. This was really important because to have these carbon credits usable on the market, you have to comply, you have to follow methodologies that are accepted already. So there's many different carbon standards that people use. What TNC went with was the Verified Carbon Standard, VERA. And they have these extremely detailed modules and methodologies that we had to go through and learn to make sure we were setting up this project in a way that if they were to move forward with it, these the quantification we got would be able to move forward and generate those credits. So one of the first steps is stratifying the site. And when I say that, it's basically carving it up, puzzle piecing the site into individual units that would have different carbon inputs. So it could be many different things for different sites, but in general, you're worried about vegetation, you're worried about soil. And then we also had a land management aspect to it. So that site stratification kind of guides your field collection. How many soil cores do you need? Where are they located? And that's sort of the roadmap to the soil coring. So we did the field collection. We collected 30 soil cores. We were also taking pore water samples for salinity. The soil cores went to the Eurofins laboratory for analysis. They're looking at % total organic carbon and dry bulk density. Those are the two measurements that you have to put in to get a picture of soil organic carbon for a site. Once those results came back, our resident blue carbon expert, which is not me, quantified the carbon, and then we reported out to TNC. So I want to talk a little bit about the stratification. We got some really beautiful glamour shots, or rather TNC did, of the site. And this shows really clearly the gradient of habitats at Port Bay. So in the upper right, you can see Port Bay, and as you go across, you see the tidal marsh, and then this, barren sandy strip, and then here's the uplands where we're expecting the marsh to eventually move up. And each of these systems is going to have different amounts of carbon stored in them just based on the vegetation and the soil and the land management. So taking an aerial view, this was our final stratification. The inputs that we had here was the Texas Parks and Wildlife Ecological Mapping Systems of Texas data. That was our vegetation classes. And then we also had the USDA Web Soil Survey for the soil types. And then the land management is represented by the thin black lines. Those are dividing up the pastures. So while we were setting this up, we didn't have that much information on how the different pastures are managed, but in case there were differences, if there's cattle grazing in one area, pooping in one area, that's its own carbon input compared to if they're not in a different pasture. Also prescribed burning, that would have a different input. So we wanted to make sure that was there. Each of the red points is where we took soil cores. In a perfect world, we would have just covered that site in soil cores, but there's budgetary limitations. Each core is going to have so many subsamples. Each subsample costs an amount of money to get analyzed. So we had to limit it to 30. So with 30 soil cores, each are in a unique combination of vegetation, soil, and pasture. That way they can all be put together at the end. So for our field collection, it was 30 soil cores between the wetlands and the uplands. The wetland cores were 1 meter deep. Our auger was 50 centimeters, so 0 to 50, and then a little deeper, 50 to 100. And you can see on the right there, that's one of the cores as it was taken out. That's divided up into subsamples based on the soil horizons that we saw in there. And that's what's transmitted to the lab. So they're looking at each subsample for total organic carbon and dry bulk density. Our upland soil cores were 30 centimeters and they were real bare. The soil there was extremely dense and hard. It was impossible to get deeper than, or sometimes even up to 30. So we had some struggles with that, which will play into the results a little bit. I'm not gonna dig into, the very many lessons learned on this, but I did wanna really highlight that it was a team effort. We had our HDR staff out there. We've got a really solid field team, but TNC was out there every day with us. A technical expert from Terra Carbon who does blue carbon work was out there with us, making sure that they were communicating best practices to us to how to get these soil cores in a way that's consistent every time. And even the landowner was out there helping pound these augers into the ground because it was a nightmare for some of them. And here's another beautiful glamour shot of Port Bay, our field team. Looking at the results, so the soil organic carbon, that's in metric tons of carbon per hectare. You're deriving that from percent total organic carbon times dry bulk density times the depth interval, how big that subsample is, times 10. Those are summed across the subsamples and summed across all the cores to get a measure of estimated average carbon for the site. So we wound up with 1,400 tons of carbon per hectare across the site on average. When you multiply that out, it was a total carbon stock of 24,000 plus or minus 2,000 tons of carbon. The results were kind of contrary to what we were expecting. What I said about the wetlands being very powerful carbon sinks, we were thinking, the wetlands are going to have higher carbon than the upland. And that's not what we saw. So we were a little confused. This is a heat map. The darker blue, it's blue carbon, so it's blue, is showing where there's more carbon, and those are up in the uplands. And you can see the marshy areas are lighter, less carbon. We were going back, we were thinking maybe it was issues with the upland soil, since we only had the 30 centimeters. We normalized it out to 100 to make it equitable across the board. We went back through the data, but we fact checked it with the NRCS rapid carbon assessment data set, and they had the same findings. They validated what we saw out there, that the uplands in these areas are expected to have really high amounts of carbon. And talking to our blue carbon expert at work, they're saying that that's not surprising for these really dense clays out there. That's something that you would expect to see. So that was good to see that, we weren't missing the mark there. So the goal for this project as a whole was really just to get that baseline assessment just of the soil carbon. If this was going to move through, you would also need to get measures of vegetative carbon. There would need to be emissions testing for how much gas is being emitted. But this was really just a first step to see if it was worth pursuing down the line. And ultimately, TNC decided that this was the wrong tool for, funding the land protection at this site. At this time, there's a lot of difficulties just in the process of working with the carbon standards and getting those credits verified. So it didn't work on this one, but it met its goal as a pilot project. I mean, we've got the methodologies down now, those lessons learned on how to make this work and what we would want to see done on future projects is much clearer now. Sort of as a take-home message on this, blue carbon or nature-based credits in general, they're offering a tool for wetland protection, for land conservation. Being able to put a dollar amount on the ecosystem services that they provide just help make protecting areas easier. 95% of Texas is private lands. That means the almost 4 million acres of wetlands we have are going to be on private lands. So being able to partner with private landowners like TNC did with the Port Bay Ranch, working with groups that know how to do this blue carbon work, makes it an accessible tool for landowners. If they're able to generate revenue on their land by doing this work, by having good land management, good stewardship of their land, and having credits on the market, that's much better for them and for us than if they're having to sell land that ends up being developed and these ecosystems are, destroyed, the gases are released. So this is still really an emerging field. There's a lot of kinks. They're still being worked out in the process, from having a project, a potential carbon project, and actually making money with carbon credits. But there's a lot of groups in Texas that are working really hard to make this an accessible tool, and that way it can be more commonplace. So I think we'll just see where the future goes on this kind of work. That's what I've got. Here's the Blue Carbon Girl Group. Really cool picture. Let me know if you have questions.
You have time for questions, go for it.
Do you foresee like assessing for like a carbon sequestration rate per year?
I was wondering about that too. I don't have that much information on the
Okay.
carbon credit side of it. That I'd like to know more on that too.
Okay, cool.
So I believe you answered this question, but do you think that that carbon would instead be found in the plants that are there instead of the soil?
It's in both. So, I mean, the soil is what's really sequestering it, right? You know, that's building up year after year after year. But there's still the carbon that's present in trees especially. And then, you know, grasslands are really powerful. They have a lot of carbon as well. So you have to assess each level to be compliant with those standards. Anybody else? All right.
Our next speaker is Adrian Hillman. Oh, yes, and next speaker is Adrian Hillman. The title is Nueces River Tidal Segment, Challenges, Resource Management, and Water Quality.
All right,
And very importantly, my co-presenter is Aaron Baxter.
I'm Adrian. Yes, thank you guys for being here. I'm gonna talk a little bit about the Nueces River Tidal segment. I don't know if you've been paying attention in the news, there's been some stuff out there lately, and so we just wanted to take the opportunity to kind of, get in front of that and get some facts out there. Maybe you guys can help spread these facts and sort of limit the disinformation that's out there. So, whoa, could you do that?
Yep.
Good job. Okay, so real quick, the Nueces River Tidal segment, if you're not familiar with it, it's about a 12-mile stretch of river. It goes from the dam at Calallen here, under 37, to where it dumps out directly into the mountain, or into Nueces Bay, bypassing the Nueces Delta. There is one monitoring station there, TCEQ has a station there. And then the segment was listed on the integrated report in 08 as having, issues with chlorophyll A levels. And then since 2020, because of fish kills that are occurring here. One thing we do want to point out is you can see on this side of the river, this is actually owned by the Coastal Bend Basin Estuaries Program. It's part of the Nueces Delta Preserve. And so it's pretty good looking, natural habitat. We want to point out on this other side of the river, though, that you've got, there's residences here. We've got industrial that occurs here. There's a wastewater treatment plant that occurs here. And so there are potential impacts to this stretch of river here. So.
And so, yeah, as Aaron mentioned, this is a segment of concern. And in part that has to do with the loadings that are actively being, that are actively flowing into this segment. But it also has to do with the fact that all water that comes down the Nueces River is controlled in some way, shape, or form. And so a brief history, there are two major impoundments on the Nueces River and a third, I would call it minor impoundment. So the Calallen Dam that Aaron referenced was built in the late 1800s and served as the first source or first water supply for the city of Corpus Christi. And also helped prevent saltwater intrusion upstream into agricultural lands. But the two major impoundments, first of which was the establishment of Lake Corpus Christi with the construction of the La Fruta Dam in 1935, followed by an expansion of Lake Corpus Christi with the construction of the Wesley Seal Dam in 1958. And then perhaps most importantly, and the largest impoundment on the Nueces River, is the Choke Canyon Reservoir, which was established in 1982 with the construction of the Choke Canyon Dam. And so pretty much any water that flows down the Nueces into the bay is controlled, and there's little to no natural flow to the bay system. And so as part of the permit for the establishment of Choke Canyon, the operators, which include the City of Three Rivers, the City of Corpus Christi, and the Nueces River Authority, were required to release some amount of water to the bays and estuaries. Back in the late 70s, early 80s, there was enough science, there was enough data at the time that regulators recognized that estuaries need freshwater inflow. And so subsequently in the late 80s and throughout the 90s and early 2000s, there were a series of agreed orders, which I have listed here. Now I could probably talk about what each one of these did and spend about 30 minutes to an hour talking through these, but I'll spare you that. But essentially, this series of agreed orders established a framework that determined target annual inflows to the bay system, established a monthly schedule that was designed to mimic historical flows, and established what's called the Nueces Estuary Advisory Council, which is a stakeholder group and group of researchers tasked with evaluating the effectiveness of these agreed orders and providing recommendations as to how to optimize flows to the bay system to maintain the environmental integrity. Coming from that regulatory and stakeholder process, there have been a couple of management efforts on the local level to, I guess, maximize the environmental benefit of those flows, which are listed here. Two of which involve diverting or piping water from the Nueces River into the Nueces Delta. And then one of them was diverting wastewater from the Allison wastewater treatment facility located on the tidal segment of the Nueces River into the Nueces Delta. Again, I'm happy to talk at length with anybody who's interested about these, but in practice, this secured some level of fresh water for Nueces Bay, but the reality is this was a great reduction on what was previously available. So in the middle of the 20th century, so pre-Choke Canyon Reservoir, the Nueces Estuary received about 600,000 acre feet of fresh water inflow per year. And following the completion of the Choke Canyon Dam, there was about a 47% reduction in available fresh water. And so as we continue and fast forward to present, obviously there are increasing demands on fresh water. Our region is, at least for this, at this point in time, almost entirely reliant on surface water for our water supply. We're seeing further reductions. This is even further complicated by periods of drought. And so here on the left side, I also have figures for inflow into the upper delta. As Aaron mentioned, the Nueces River now bypasses the delta entirely. And so one of the management measures that were recommended by the NEAC was to allocate some amount of water to the Nueces Delta, as that would have a greater environmental benefit. But tying this back to the Nueces River tidal, remember you have a saltwater barrier up in Calallen, which means that everything below that is entirely reliant on spills over that dam into the tidal segment. And so this significant reduction in available fresh water leads to elevated or increased residence times. And when you have inputs or pollutant loadings, that can create some issues. I'm not suggesting that dilution is the solution to pollution, but flushing certainly helps the water quality in this segment. So in practice, even with these reductions, I just wanted to share this. This is the only figure I think we have in the presentation for a reason. But so the way this works is every year there's a target amount or volume of water, which is 151,000 acre feet. That amount is reduced based on certain criteria, whether that be lake levels, whether that be current bay salinities. But in practice, there are essentially three main mechanisms for water to arrive to the Nueces Estuary. You have spills over the Calallen Dam, which is this orange line here. You have what are called return flows, which is a calculated set amount that the operators are credited, which is 54,000 acre feet. That's this light blue line. That's essentially all water that is used or consumed by people that then eventually makes its way back to the bay system. So think wastewater discharges, any use that is then returned into a river segment and into the bay system. And then you have this green line, which is the piping of water to the upper Rincon Bayou. But so what I want to demonstrate here is that the volume of water that reaches the estuary fluctuates greatly. And in practice, this agreed order, while it does deliver some amount of fresh water to the base system, what ends up happening is we end up getting inflows when there is an abundance of water. And there is little to no water available for the estuary when it's needed most. And so you can see in 2015 and 2018, very wet years. I think we had in 2018, something like 180 days of water spilling over the dam. But since then, we have not met that 150,000 acre feet target of inflow to the estuary, which established by the agreed order. And so that is an important component and an important thing to understand as we talk about loadings and water quality issues in the Nueces Tidal. But the way that these inflows are regulated and work is also a very charged topic amongst the public and a very misunderstood point of conversation, where we have conversations with residents along the river who are under the impression that water is released at all times or not at all, or that all the water is being pumped across into the Nueces Delta. And so it's really important that folks understand how the water is regulated and how it actually works when being implemented.
One more thing to note, when the lake levels get below a certain level, they don't pump any water. And so that's why there's no water that's come down the Rincon Bayou into the Delta in 2 years.
So yeah, to add to that, so as lake levels decrease, the target amounts in a given month or a given year are reduced. When lake levels hit 30%, all water releases for environmental purposes ends. The catch there is, regardless of lake levels, the amount that is released or passed through to the estuary is capped at what they call the measured inflow to the reservoir system, which is a measure of all the water that flows into the system minus the permitted releases for human uses. So everybody who owns a water right, they release a set amount of water, and so the water that is available for the estuary is total inflow minus the permitted releases. So let's say there are 100,000 acre feet of water, of permitted water that are released every month for human uses. We need 101,000 acre feet of inflows to have any water available for the estuary. So even when we're not in drought, if there is very little rainfall in the watershed, specifically up north of Choke Canyon, there's very little quote unquote inflow to the reservoir system.
Great. So now we're going to talk a little bit about some of the environmental concerns in the Nueces Tidal segment. One thing to note is that these tidal rivers, creeks, these are naturally stressed systems to begin with, right? I mean, between a freshwater and saltwater source. And so, you can have big spikes in salinity. You can also go fresh if you've got a flood, from, say 25 parts per thousand to 0 in a matter of hours if we get a big flood. And so these are naturally stressed environments to begin with. And then you reduce that freshwater inflow and they can become even more stressed as a result of that. And so we had some residents, like Adrian mentioned, some of them are quite vocal. They're all very passionate though, and this is where they live. And so it's very, very important to them. And so they came to us a few years ago with some concerns, mostly related to algae blooms. If you've ever been on the Nueces Tidal, there's a picture of water from the Nueces Tidal segment. It's usually pretty green. Fish kills, here's a picture of a large fish kill that occurred on the Nueces Tidal. And then contract recreation, is it safe for them and their kids to get in the water? And so these are all very valid concerns. We took it very serious when they came to us. And, we want to start looking into it. So we got with Alex, Parks and Wildlife Skills and Spills team, just some basic, fish kill data here that he provided to us. One thing we do want to point out that over those 73 to 2013, it's a 40-year period, there was 15 documented kills. Between 2018 and 2024, that's just a 6-year span, there was 9. And so without getting too much into the weeds on this, it appears that the frequency of fish kills has definitely increased along that stretch of river. It is interesting to point out that there was none documented in those 3 years there. That's why I did not include any numbers there. And the majority of these events have been attributed to low dissolved oxygen events. And so you want to go to the next one there? So what are we going to do about this? We know we've got some issues out there. We've got to think of ways to address these issues. And so we're going to go ahead.
Well, I was going to say, one of the things, and Aaron touched on this, one of the things we've realized very early on is there was very little data, water quality data, for this segment. So there was quarterly monitoring by TCEQ, mainly for chlorophyll. But aside from that, there was no higher resolution monitoring, spatial temporal monitoring on that segment, looking at nutrients and bacteria.
That's a great point. I mentioned earlier that TCQ has one station there. That's really about all the data, you know, that was being collected outside of, say, Alex gets a call, his team's got to go investigate a kill, something like that. But so yeah, we got together with a bunch of residents that live out there, representatives from the city of Corpus, from Nueces County. We tried to form this really diverse stakeholder group. We organized that officially in January of 2023, and we wanted to model it after the very successful Baffin Bay stakeholder group. You know, they're making some really significant changes down there, community-driven changes, and so we thought that would be a really nice model for us to use here. And so we met with these folks. The initial meeting was quite heated. I think some of them might have been intoxicated. It was it
5 o'clock, you live on the river.
you know, I mean, I don't blame them. I would have been too if I wasn't at work. But so we take Dr. Wetz out there, you know, we really helped lead that stakeholder process down in Baffin. We brought him on board, and honestly, after our first meeting, Mike said, "I don't want to do this." He said, I'm not dealing with these people." Fortunately, we had one resident out there that was willing to sort of take up, you know, the charge, lead the effort, and he's done a very, very good job of getting them to just calm down a little bit, realizing that we're on their side, you know, and so things have gotten better. Our relationships have improved. We've had 2 other stakeholder meetings since that initial one, and so things are trending in the right direction. So great, we got everybody together, we're listening to their concerns, and then we've got to think of ways, how are we going to address these concerns? And so we funded 2 studies so far in the last 2 years. The first was led by Dr. Mike Wetz. Oh, geez, I'm done. All right, so nutrient bioassays are real quick. It looks at what nutrient is possibly driving these algal blooms that we're having, right? The algal bloom eventually leads to the low DO, which leads to the fish kill. And so what Dr. Wetz showed is that we've got a nitrogen issue. He looked at nitrogen, phosphorus, and a combination of nitrogen and phosphorus. It looks like nitrogen is the driving nutrient here. And then Sam at the Center for Coastal Studies did some bacterial stuff, some just other water quality characterization. Then obviously our next steps in finding out that nitrogen is probably what's driving this, we're looking at maybe doing a source tracking study for where is that nitrogen coming from. And then also I'd like to look at, you know, a bacterial source tracking as well to find out where these high fecal bacteria numbers are coming from. I think I'm out of time. So, hey, look at that. It's not that bad.
Yeah. Report now, and yeah, we're excited to present some results to that stakeholder group in the near future.
We got time for one quick question.
That's good.
Okay.
So Adrian, that's great information. I'm wondering whether to reduce the flow, whether affecting how much sediment is being transported. You know, you're thinking about maybe 30 years, 50 years, whatever. Is any motivation to look at the landscaping changing with this time frame? Because you're talking about the sediment transport, but also the sea level rise. Wondering how I know there's a lot of channels, right? So how the morphology, is that changing or?
The morphology of the Nueces Delta is changing drastically and rapidly. So Ken Dutton here at UTMSI has been working in the Nueces Delta for a long time. And I believe the estimates are that we're losing about 15 feet. Lee, is that correct? 15 feet per year. So the bay and delta face of the Nueces Delta is retreating about 15 feet per year. So it's significant marsh loss, significant habitat loss. We've just been working with the Army Corps of Engineers to place beneficially used dredge material to try to restore some of that lost marsh. We've also constructed some breakwaters to try to protect the delta face and reduce and slow erosion. But that's the other side of the coin. When you have reduced inflows, you don't have as much fresh water, less mixing, but the sediment transport is an incredibly value component that is greatly reduced and lost.
Yeah, so I'm saying there are a lot of nice remote sensing data. If the resolution enough, maybe it's useful to do a comparison, you know, this time, how that been changing. So you get some quantitative data. I don't do it, but I think that's probably something. Anyway.
I think that's a great idea.
And if you're interested in learning more about the Nueces dredge placement, Lee and I will be presenting at 2:00, I believe, on that. So if you're interested, we'll be getting more into that.
Yeah, it's more of a comment than a question. I think that So whenever we talk about these water quality concerns and stuff like that, Parks and Wildlife, by the way, Alex, is. One of the things that we worked on this was using that same model as Baffin Bay is it's easy to point the finger at a point source or some other group. But without the data, it doesn't support anything. So doing this like data gathering is really important to us find the solutions because it really is based on building partnerships, not pointing a finger and accusing folks. It's how can we work together to resolve this issue? And I think that's one of the biggest things, takeaways for this, as a lot of y'all are moving forward in life. That's the biggest thing is not the accusing part, but the building of partnerships. So thank you, Estuary program, for taking this lead and you know, all the work y'all are doing to address this issue in the tidal sector.
Great. Thank you. All right, we have one more speaker before we move on from lunch, and that is David Essien. And his title is Quantifying Habitat Loss at Waterbird Colony Island.
Thanks for coming to my talk, everybody. And when I think about breeding waterbird habitat on the coast, I think about coastal infrastructure. We all rely on coastal infrastructure as residents here on the coast. We need things like seawalls, bulkheads, breakwaters, and navigation channels to go about our daily lives. But as coastal ecologists, I think it's important to also think about how these structures change the physical environment by altering native habitats and also creating novel habitats. And they alter functional diversity patterns as a result. So they affect what species can live where, species composition, and relative abundances of species. And also, it's important to remember that because unlike natural reference habitats, novel habitats aren't created or maintained through natural processes, they have a lifespan. So as soon as they're constructed, they start to degrade and eventually they become really unsuitable as habitat. And we're nearing the 100-year anniversary of the construction of the Gulf Intercoastal Waterway, which is really a centerpiece of coastal infrastructure on the Texas coast. It was constructed from 1928 to 1949. And after 1949, it was really just maintained through maintenance dredging. So during the process of construction, over 300 islands were created. There are these spoil heaps along the channel. And almost immediately, coastal waterbirds started colonizing them. So over 20 species of waterbirds began to use them as nesting habitat. And now they support over 100,000 breeding pairs of waterbirds per year. So certainly they affected functional diversity of waterbird communities on the Texas coast by changing the nesting habitat from a system where there were just a few large islands that were kind of spread out along the coast to a system where there's a whole lot of islands that are a lot more connected, and many of the islands are a lot smaller. And also, again, because these islands were created by humans and aren't really maintained by natural land building processes like sediment deposition and accretion, at least not at the level that can keep up with erosion and sea level rise, they're eroding rapidly. And managers have realized that this is a potential threat to waterbird populations in the future. And so they've started to rehabilitate habitat by sometimes building new islands near where islands had previously become submerged, and sometimes by maintaining islands with dredged sediment from nearby channels, like you see in these photos. But there are too many islands and it's too expensive to do this at all of the islands. So they're really looking for ways to prioritize which islands they rehabilitate. And that's a big part of the project I work on. And they're also sort of lacking information on rates of habitat loss. And that means they don't have a really good understanding of how long until these islands are no longer suitable and how it's going to affect bird populations in the future. And so that's really the objective of this study, is to figure out how fast islands are losing habitat, what types of islands are losing habitat the fastest, and how water bird populations are responding to habitat loss overall. And so to do this, we delineated imagery from the National Agricultural Imagery Program, or NAEP imagery, from 1995, 2008, 2012, and 2018. We delineated vegetated and unvegetated habitat at 223 islands from Redfish Bay south through the lower Laguna Madre. And we did that because there are birds that nest on bare substrate on these islands, and there's birds that will nest on vegetated substrate. You can see on the map on your right that the islands in the left panel are represented in green. They're really hard to see because they represent a really small proportion of the landmass in this region. On the right panels is Shamrock Island in Corpus Christi Bay, Pita Island in the upper Laguna Madre, and Green Island in the lower Laguna Madre. So those are just examples, and you can see the different shades of green. Green are delineation from each year of NAEP imagery. And I also want to point out that this is a really sparse time series that we have imagery for, and these islands are changing constantly. So we are leaving out a lot of information, a lot of variation in the area of these habitats in this analysis, and that's just a limitation that we should keep in mind. Okay, so what do we find? We found that habitat loss was rapid and widespread. So vegetated habitats decreased at about 2% per year at 77% of our islands. And unvegetated habitats decreased by about 3% on average per year at 86% of the islands. But we wanted to take this a step further and ask what types of islands are losing habitat the fastest. And to do this, we use hierarchical Bayesian models. Really, the idea here is that we wanted to be able to kind of predict the area of a given habitat in a given year as a function of the site, the year, and the rate of loss. And we wanted to be able to predict the rate of loss as a function of different island traits. And so these two submodels are linked. That's why it's a hierarchical model. The covariates that we use to model habitat loss rates are island area. We predicted that smaller islands would erode faster because, if nothing else, they erode from the perimeter in, and smaller islands have a larger perimeter to total area ratio. We predicted that the fetch length would influence habitat loss rates. So that's an indicator of wave energy. So the farther wind has to travel across the body of water, the more energy, more wave energy can be generated. We predicted that lower elevation islands would erode faster, and islands with lower percent vegetation cover would erode faster because vegetation cover can provide some wave attenuation and also stabilize sediments. So, and what we found was that smaller islands did erode faster. So that dash line at 0, basically the line below the dash line represents habitat loss. So negative numbers mean that that the area of habitat was declining. And we also found that islands with lower percent cover lost habitat at a faster rate. Okay, so our waterbird populations responding to habitat loss at the regional scale. We already know that they respond at the island level. There's been reports by coastal bend basin estuaries in Mesquite Bay that show that islands have lost habitat really rapidly, and at those islands, bird populations have declined, which isn't surprising. But we also have to remember that there are over 300 islands in the system, and birds don't use all of the islands in every year. And they can move from an island when it doesn't become suitable to another island. And so these colony dynamics can provide some stability to the population. So we fit dynamic occupancy models to estimate colony persistence. So what's the probability that islands will persist from one year to the next? Expecting that if habitat loss is affecting colony dynamics, the probability of islands persisting from one year to the next overall in the system will decrease over time. And we also just examined population trends of the aggregated populations of bear, substrate nesters, and tree nesters. And we found that colony persistence didn't really change over time. So basically the probability of birds nesting at an island from one year to the next didn't change overall. And the number of active colonies didn't really seem to change overall either. And we found that the number of breeding pairs of ground nesters and tree nesters didn't decrease. I split these up, so I aggregated all of the nests of ground nesters and tree nesters at core and peripheral islands. The core islands are the few islands that support most of the breeding pairs. For tree nesters, that's only 3 islands in our study area support about 60% of the nesting pairs. And for the ground nesters, it's about 9 islands in the study area that support about 60% of the breeding pairs. The reason I split those up is because I didn't want the core islands to sort of swamp out effects that were occurring in the peripheral islands, which tend to be smaller. And the smaller islands are eroding faster. So in any case, we didn't find any decline in numbers of breeding pairs being counted at these islands over time. Okay, so when you really take a step back and look at this, I mean, we have habitat loss rates of about 2 to 3% per year on average in our study area. So if you look forward, even just 40 years, that means that we're not going to have as much habitat as we had, obviously, but we're losing entire islands even. So we've already lost, during the study period, 31 islands that became completely submerged. And our models predicted that 51 more will likely become submerged within the next 40 years. So that means that the network of islands that these birds nest on will become smaller, and the islands that they're able to nest on will have less habitat than they do now. But the good news is that bird populations haven't started to decline yet, and managers are already taking action to rehabilitate some of the islands that are losing habitat most rapidly. So that I just want to acknowledge the interns, especially those 3 names at the top who did all of the manual habitat delineations in this study, because that was a lot of work and a lot of time, and then also our funders and collaborators.
We got time for questions?
So you said that the bird populations haven't really decreased, but the islands have gotten smaller.
Yeah.
So is there any issues that come from birds being closer together? So any issues with crowding on these islands?
Yeah, that's a good question. So this is really one small part of a larger project. And in the larger project, kind of getting to your question, we're monitoring nest survival. And that's kind of the first thing that comes to mind when you mention like effects of birds being crowded. You know, maybe you're right, maybe there's an effect on probability of survival or the fitness of these birds as a result of habitat being, you know, less suitable, maybe not so unsuitable that they can't nest there, but less suitable. Yeah.
Great presentation, thank you. I wanted to ask, I know your study area focused on Redfish Bay and South.
Yeah.
Would you happen to have any sense of, given that, you know, more north, there's like higher erosion rates along the coast?
Yeah.
If there is maybe a sense of how many islands would be remaining or actually submerged, I should say.
Yeah, so we have interns delineating habitats for those islands right now. So yeah, hopefully we'll have a better idea. And also, some of the work has been reported by the Audubon Society, like they've done shoreline retreat analysis on islands further north. So that information is available somewhat for a shorter time period. So yeah.
Do the islands ever receive like new dredge material when they dredge the ICW?
Some of the islands do. I tried to filter out islands that have been restored or that I know have received, you know, maintenance, dredging, spoil. But yeah, most of the islands, I'm not aware of any that were included in our study area that during the study period received, you know, a whole bunch of dredge sediment that would have increased the area of habitat. It's kind of hard to get that information. I've tried to contact the Army Corps of Engineers to get like dredging schedules or like really, I want to know the history of, just like you're saying, dredge placement at these islands. And it's, you know, it's difficult information to track down. So.
All right, well, thanks everyone for coming. We do have a substantial lunch in the Patton Center. And then please bear in mind that they still have a talk going, so try not to disturb them if you can. Thanks so much, great lunch.
One day.
Like, down by the lake, you know, it's about to see
To re-identify dredge placement areas, they may not be, available to be placed there because of the rookery season.
Thanks, buddy. I appreciate that. Yeah.